in the 20th century, muscles reigned. The most famous development came from this man, Wilder Graves Penfield, seen here as a young man and then in his more distinguished era. Most famous is his artist's depiction of his work with Bouldry in 1937, seen here in its early form, and finally in 1950 with Rasmussen in its well-known form, the homunculus, which literally means little man. A picture is worth 1,000 words, but it was good research, compiling data from 163 fully awake brain surgery patients. His work described muscle twitches rather than the complex movements reported in the 19th century by Ferrier. However, Penfield and Welch noted in their 1951 paper that it is our impression that many of the postural synergies which result from stimulation of the supplementary motor area would, with more prolonged stimulation, have gone on to become more elaborate and to have approached the full synergic employment of all extremities and the trunk. And the stimulating electrode has frequently been removed at the first evidence of response, and thus the opportunity of producing more of the elaborate synergic responses may have been missed. If this surmise is correct, it would be proper to consider many of the motor effects as fractions of an uncompleted complex. There are three other notable research directions in the 20th century that greatly impacted the understanding of motor control. First, in 1966, Schick and colleagues stimulated a locomotor region in the midbrain of a decerebrate cat, and the animal walked with support on a treadmill in a normal fashion. Interestingly, by increasing the strength of the stimulation, the cat's walk progressed to a trot and then a gallop. Whilst the animal did not do well with obstacles and could not stand alone, this research demonstrated that regions other than the motor cortex were involved in motor control, especially locomotion and other rhythmic functions like swimming, flying, respiration, and swallowing. A neural network or even a single cell producing a rhythmic motor behavior without sensory feedback is known as a central pattern generator or CPG. That locomotion can be produced by the spinal cord is well known because of the headless chicken running and flapping its wings. One such headless chicken called Mike lived for 18 months. This research has been extended to the motor control of locomotion in various vertebrate species, including lampreys, salamanders, stingrays, rabbits, and monkeys. Jumping ahead into the 21st century, briefly, Elke Ishpiet, a Swiss roboticist, has developed a robot based on the neural control of the salamander. Let's hear from him.
So we studied salamander locomotion in more detail, and we had in fact access to a very nice X-ray video machine from Professor Martin Fischer in Jena University in Germany. And thanks to that, you really have an amazing machine to record all the bone motion in great detail. And that's what we did. So what you see here is almost a direct comparison between the walking of the real animal and the robot, and you can see that we have almost a one-to-one exact replay of the walking gait. If you go backwards and slowly, you see it even better.
And secondly, the other most notable occurrence during the 20th century was an advance in technology for cortical stimulation. Intracortical microstimulation was introduced in 1967-68 by Asanuma and colleagues, probing the depths of the motor cortex, providing a more localized stimulus even down to the single neuron, and going beyond the limitations of the more superficial surface stimulation of the past. And third, transcranial magnetic stimulation, or TMS, was developed by Merton and Morton in 1980. TMS applied to the motor cortex evokes muscle twitches and even functional synergies in the muscle groups related to the area being stimulated. Research of motor control with TMS continues, but it has also found a place in treatment of various neurological and psychiatric conditions.